Hello everyone, we are live on YouTube. Thank you for your patience.
Shall we start the session?
Absolutely.
Okay, great. So, good morning, good afternoon, and a very good evening to everyone. Welcome to this third day of a 5-series webinar workshop where distinguished experts are sharing their experience on taking novel drugs from lab to clinic. This event is organized by Drugs for Neglected Disease Initiative, DNDi, in collaboration with BIDAC and ISCR. In the last 2 sessions, we have covered the framework of successful lead optimization and the fundamentals of pharmaceutical development. And in continuity, today, our esteemed chair and panel of top experts will elucidate on Pkpd relationships and safety pharmacology. But before we move forward, I would like to introduce some ground rules. So first, please change your names on your laptop or your computers to the one that you have used for registering to this event. That is to say, your name on your screen should appear the one that you have used for registration. And second, please keep your microphones on mute. That would allow and facilitate a smooth session. And with this, I would like to introduce our session chair, Dr. Sridhar Narayan. Dr. Sridhar has more than 20 years of drug discovery and development experience in Indian pharmaceutical industry in various therapeutic areas. He holds a basic degree in pharmaceutical sciences from University of Mumbai, a PhD in pharmacology from Ohio State University, and has a postdoctoral experience in neuropharmacology from the University of California, Los Angeles. Dr. Sridhar is a serial entrepreneur. He is currently founder, director, and chief executive officer of Foundation for Neglected Diseases Research, FNDR, a not-for-profit company with a mission to discover and develop drugs for diseases of the developing world. With this, I hand it over to Dr. Sridhar. Dr. Sridhar, over to you.
Thank you, Soman. Welcome to all the participants. I'm sure you've had a wonderful learning experience over the last 2 days in lead optimization and pharmaceutical development. Today's session promises to be another blockbuster, focusing on the very important topic of Pkpd relationships and safety aspects of drug development. High attrition rates during the drug development remains a critical issue. We must address a series of steps to better understand the pharmacokinetic and the pharmacodynamic properties of novel agents to increase the probability of success in drug development. With the increasing volume and complexity of data that is being generated in the preclinical research, a comprehensive integration of pharmacokinetic and pharmacodynamic information into predictive mathematical models is the need of the hour. The utility of Pkpd models extend not only in identifying and selecting the best clinical candidates, but also predicting clinical response in humans and facilitating understanding of the potential relevance of preclinical efficacy, safety, and toxicological data in a clinical setting. During the clinical development, Pkpd modeling and safety can help optimize the design of clinical trials, guide the dosing regimen, and also the dose that should be tested in the volunteers, help anticipate the effect in certain subpopulations, such as a geriatric or a pediatric population, and better predict drug-drug interactions, which lead to a much more efficient drug development process. I'm honored to be chairing this session and have a speaker panel who are experts in the Pkpd modeling and safety testing. It is my pleasure to introduce our first speaker, Dr. Ayman Al-Katan, who's the Executive Director and Head of DMPK at IFM Therapeutics. Previously, he was Associate Research Fellow at the Pharmacokinetics, Dynamics, and Metabolism Department at Pfizer, Cambridge Laboratories, where he worked for 17 years. Dr. Ayman has a bachelor's degree in pharmacy with distinction from the University of Jordan and a PhD in basic pharmaceutical sciences at the University of South Carolina in the US. He is also an adjunct professor at the College of Pharmacy, University of Rhode Island in Rhode Island, USA, where he lectures in the graduate level pharmacokinetic courses and serves as an external advisor on dissertation committees. Ayman is an active member of the American Association of Pharmaceutical Scientists, and serves on the committee of the Drug Transporter Community. Dr. Ayman has also published a book titled Oral Bioavailability Assessment, Basics and Strategies for Drug Discovery and Development through the Wiley Press. I welcome Dr. Ayman and look forward to an exciting talk from him. All yours, Ayman.
I want to first share my slides. I want to confirm. Can you guys see my slides before?
Yes, yes, Ayman, we can see your slides.
Excellent. So
you might just want to put it on. Yeah, fantastic. Thank you.
So first, I would like to say a very special thanks for our friend Dr. Narayanan for giving me this great opportunity. I'm really honored, and I'm really looking forward for a good discussion on this emerging forum. When I was talking to the organizing committee, they have emphasized that many of the attendants are graduate students or postdoc. Before I'm going to start, please, if you are doing work in the area of pharmaceutical, drug discovery and development, put a lot of focus on the area of Pkpd. The opportunities that are in this field are amazing, and we are always looking for good talents, especially people coming from India, but don't tell the Chinese, by the way. Especially with your strong background in math, it will give you an edge that you really need to leverage. Okay, so now why don't we start talking about this exciting concepts of PK and Pkpd, and see basically what are the basic principles of this important field that is critical for the success of any pharmaceutical institutions. The outline of this presentation is going to be as follows. First, we will start with where we are going to be discussing and providing an overview of Pkpd principles. After that, we are going to be talking about why, where we are going to be giving examples. And then we're going to share with you a roadmap for how we can put all of this in perspective. The presenters who are going to come after me, they're going to give you more hands-on, in-depth examples that's going to help you to have really a good grip on this exciting concept. Okay, now, when we are thinking about the Pkpd, we need first to start with the very first one, which is the concept of PK. Now, what does PK stands for? The PK, it's very simply, it is what the body does to the drug. And when we say that, basically, it is the principle that will describe 4 key processes for any drug that gets administered into the body. And basically, these concepts are the following. Number 1, absorption. So in terms of absorption, it is basically a description of how the drug will be moving from the site of administration inside the body. So for example, if it is orally, it is the process that will describe moving the drug from the site of administration to the inside of the intestine. So that's by itself is a discipline. The second discipline, once the drug is absorbed, of course, it's going to distribute. So that means it's going to go from the intestine into the blood, then the liver, and then it will go into the whole circulation. That process has a lot of mathematical principles that can be that are important for us to put these things in perspective. After that, we are talking about the drug is not going to stay there forever, because what will happen, the body is going to start undergoing a process which is called clearance, as the name indicates, clearance. So basically, you're clearing the body from this drug, and that is going to be an interplay of various factors that are going to decide how the compound will be eliminated. The last process that happens when this is going to occur for the drug is going to be the active process of excretion. And when we say excretion, what do we mean? It's basically the complete elimination, where the compound is going to be completely exiting the body. That can happen by an active secretion in the bile, or it can happen by an active secretion of the compound in the bile as a form of metabolites, or it can be by elimination through the renal route or the kidney into the urine. So now, so basically, this is the principle of the PK, which is what the body does to the drug. Now, of course, when we have all of this, what you are going to see is you typically will see this simple form of a relationship, which is, the drug plasma concentration versus time. So basically, on the x-axis, you have time, on the y-axis, you have concentration, and this is how you describe all of this. Now, the PD, this is where you see I have witnessed a major change in this discipline. Back in the old days, the emphasis was taking, assessing the impact of doses on an in vivo marker. And then they're going to be doing the scaling. But things are getting way more complicated than when I started. What you start observing is that the people are putting a lot of emphasis on using modalities such as biomarker, efficacy, toxicity, and more important, relating these changes in these parameters, which are the biomarkers, efficacy, in vivo efficacy, as well as in vitro, and toxicity, to drug concentration. So now this is just a nice example for a simple sigmoid Emax model, where you set a relationship between the drug concentration and the effect. And as you can see, the more you will see your drug profile on the left side, the more potent it is compared to the least potent. So this is really a very exciting concept that I think we should put a lot of emphasis on. Now, so again, as in a form of how things are changing, back when I started in the drug industry, the PK and the PD, they were not, always, the tie was not really strong. But you start seeing changes when we start putting more engineers in our discipline, and we start getting more biomedical engineers, and you start seeing more math. What you start seeing is a healthy relationship between characterizing the drug dispositions in PK and also how it's going to affect the drug target. This is actually where you started to see more investment of math to put all of these things in perspective. Now, so what we are going to be doing today is we are going to be talking about understanding the relationship between drug concentration and effect versus time. And all of that is going to be done using modeling approaches and mathematical tools that are available at our discretion. I need to share with you some good news. We're not going to be talking about a lot of mathematical models. I'm not going to, put that pressure on you, but we're going to still show some diagrams, so nothing is perfect. Okay, now let's start talking about the models that are available for describing a Pkpd relationship. The very first model that we're going to be talking about is a direct effect model, as the name indicates. The beauty about this model, which, by the way, in pharmaceutical industry, we get really happy when we see that we're just going to be stuck with a simple direct effect model. It's not going to be complicated. It's going to be a cruise sailing. So over here, what happens is very simple. You are going to be administering the drug orally, okay? And when the drug is going to be administered orally, it's going to be absorbed. Once it's absorbed, it's going to be available to interact with the site of action. The site of action can be a target, and based on the target modulation, you're going to see a drug effect. So now the drug effect, how much is going to happen in terms of drug modulation? It depends on how much drug concentration is available to basically end up with this needed biological action. So now, as a result, we typically, when we establish this kind of relationship, we actually use a direct effect models or an Emax model to describe the relationship. And the beauty about this, you can easily put the PK in perspective. The PD, you just understand the changes related to the drug concentration, and voila, you have the target concentration that you would like to achieve. And based on that, you can do all your modeling and simulations to make up projections for the human dose, which is really good. And also, it's very easy to be done. Now, but the problem here is it's not always easy. You don't always, you're not, especially lately, we are actually embarking on much more challenging models to describe drug activity. And also, we are targeting much more complicated approaches to basically modulate the drug target and end up with efficacy. So that's where we start having that. Sometimes what you will notice is that you will give the drug concentration. And what's interesting, look here, this is the same time profile, but you notice is that although the drug concentration is low, you're still seeing efficacy. So what you are actually seeing, a delayed and sometimes a prolonged effect. I remember the first time that I saw that, my background was in PK, and I didn't put a lot of emphasis on PD in my training. So basically, I learned that this on the job. Like at that time, we started asking, like, what's happening there? Why we are seeing this trend? Because most of the models that we were working on, they are basically direct effect model, which is very simple math compared to this. We start seeing that actually the relationship, we call it hysteresis. And this hysteresis is where you're going to see a delayed or prolonged effect. And it is much more complicated and it requires much more math compared to the direct effect model. Now, here is actually a work that a compound that we worked on in Pfizer. we were working on trying to come up with a better antipsychotic agents. It was a me too. That was the time when Pfizer was still investing in me too compounds. We wanted to provide a molecule that is better than aripiprazole. This is when we start seeing these kind of relationships, and we start seeing that the concentration relationship with the biology is actually not linear, not direct. And this is where the excitement started to happen, and people start saying like, oh, we can start making use of this. So now, hysteresis, I remember when we were in Pfizer and we were starting to say that there is hysteresis, that was not always a good news from the fact that it's going to require way more math. But at the same time, it has a lot of advantages. As a matter of fact, like this concept of hysteresis is actually one of the main one of the topics that's being used to describe, for example, covalent binders and also protein degraders, because we start using modalities that it was like back in the old days a taboo for using. So this is actually really exciting because it ended up broadening our perspective. Now, what are the potential causes that will end up having us to have an indirect model or a hysteresis? Let's go through it. So first of all, you have an active metabolite. As you know, the active metabolite needs first to go into the liver. There's going to be a generation of the metabolites, and the amount of metabolites generated is going to dictate how much activity you have. So that can be a potential. Sometimes slow distribution effects, basically, the distribution of the compound into the site of action is not as fast as you would like to see. Also, you have a tight receptor binding, and the biological turnover, regardless if it is increased synthesis or a decreased elimination, that can play a role in the delay of the compound and prolonging the compound activity. And of course, signal transactions and all that kind of stuff. So as you can see, the exciting part about this indirect model or hysteresis, it helps you to dig deep in the mechanism of action. And guess what? Having a solid understanding of the mechanism of action is a prerequisite for being successful. And as our Dr. Narayanan mentioned, it's going to reduce the odds of you having attrition, which is still high. So now, what's the take-home message? If you're going to be in drug industry and you're going to be responsible for the Pkpd, and you are starting to investigate drug concentration versus effect over time, you need to always ask yourself, am I seeing hysteresis or not? Because if you do, or even if you don't, understanding that will help you to understand the mechanism, and as a result, it will help you to put these things in perspective. Now, why Pkpd? Again, we're going to emphasize on what Dr. Narayanan emphasized. What did he say? He made it very clear that the attrition rate is high, especially in the beginning of this century. We had a very high attrition rate, and the return on investment was not as good. I remember one time I have heard that the CFO of Pfizer, that was in the beginning of this century, he was saying, I will be making more money if I'm going to put the investment I make in research in a mutual fund like S&P 500. But of course, that was a very strong message from a CFO, which actually pushed Pfizer to really work hard to make sure that their investment is going to be better in terms of work and in terms of reducing attrition. And I think Pfizer is doing a much better job compared to before. Now, why we are doing preclinical Pkpd? When you have a preclinical Pkpd, what's the beauty about it? You're going to have a solid understanding of the drug interactions with the target over the course of the time. What's the benefit of that? Based on this, you're going to be able to decide what is the efficacious exposure. It's going to help you to have an idea about the safety margin of the molecule and how many times you're supposed to be giving your drug in terms of your population. So is it going to be once daily, BID, and how big is the dose and the cost of goods. So this work that we are doing in preclinical PKPD, it has a huge impact down the line. So that's why it's important for us to make sure that we are putting it in perspective and we have a solid understanding of the drug dispositions overall. Again, let me share with you a two perspective. As you can see here, the red bullet is the Pfizer X compound, and the blue bullet is aripiprazole. If you look at the relationship between dose and the percent of inhibition of the locomotor activity, you will say, hey, are you kidding me? Like, Pfizer molecule is much better than the aripiprazole. As you can see, there is, you see like that the ED50, it's going to be around like, let's say, let's say 5, and over here it's around, let's say 4. So aripiprazole for sure is less potent, but that's not right, because when you look very closely at the plasma concentration and the relationship between plasma concentration of aripiprazole versus the one in Pfizer, you will see superiority. So that's why you see this was the old era of the 90s where we used to put a lot of tokens on the dose. This is the new where you are putting an emphasis on a relationship between concentration versus activity, and this is where the money is. Again, now the beauty about the PKPD, we don't just stop with EC50, like let's say, or EC90. It is much bigger than that. What you are seeing lately is that now people are adding also a confidence interval. What does that mean? That means when you are going to be giving an EC50 target, you are going to emphasize that my confidence interval of that value is within this range. Of course, the smaller it is, the more confidence you have, and you are seeing more emphasis on putting all of these things in perspective, which is great. Now, when you have a better estimate of drug potency and durations, of course, we are not just doing that from a single dosing, we're also doing this at steady state, because sometimes what happens when you give the compound multiple times, you may see an induction that's going to see a change in the concentration, and the behavior of things will change. So that's why it's important to do steady state. Okay, so now, again, PKPD is a quantitative drug discovery and development. So as a result, what we are looking for is we are looking for fine students whom they have a solid understanding of biology and also a solid basis in math. I'm going to take this opportunity by saying, if time will go back again, and I am going to go back in time and I'm going to study, other than what I have studied, I will make sure that I will get a master in statistics, or I will add more courses in math. I cannot stress this enough, and I hope that the message has arrived. Okay, now, the beauty about PKPD, it is a teamwork. What does it mean being a team? What it means, it's very, very simple. Together, everyone achieve more. Now, in drug industry, I cannot stress enough that the success of you as a drug hunter depend not only on your scientific knowledge and your competency, but also in how good you are in working in a team setting. The era of one-man show is over, and you need to know and master how to work in a team setting. And being in a PKPD is really a prerequisite for that. Because guess what? You're going to be working with DMPK scientists. You will work with biology, clinicians, clinical pharmacology, safety, translational research team at large. This is going to be the basis for ensuring the quality of your projection. Now, I want to stop here and ask Dr. Narayanan how much time I have.
Ayman, you have another 7 minutes.
Excellent. Okay, so I have time. Thank you. Okay, thank you, sir. So now let's talk a little bit about the roadmap and how we are actually approaching the drug, the PKPD, and putting those in perspective. what's going to happen is you're going to go through each and every one of these steps to end up with an accurate, meaningful projections of the efficacious concentration. So now let's go into each one of them. First of all, our chemists are going to start in screening, and they are going to do initial compound screen. Now, in this phase, what happens, our chemists, they are going to end up putting certain targets that they are supposed to be achieving. And when I say target, what do I mean? For example, now in my current work in IFM, our chemists are going to have a set of in vitro biology assays where the compound is supposed to be hitting them to show that the compound has a good potency toward the target. That's number one. And number two, it is selective. What does it mean, selective? That means it is just hitting the target that it should hit, and it's not mingling with other targets that may affect and reduce its efficacy. Now, in addition to all of that, they're going to also have an in vitro ADME studies that will show that the compound has a good likelihood of having a good stability from a metabolic standpoint, and the compound has a good basis for being absorbed. So once we do that, what's going to happen? We're going to start designing the right animal studies. So typically, what we do is we're going to identify a dose. It's typically a single oral dose, and what we're going to do is we're going to be collecting plasma profile and plasma PK to make sure that the compound has a decent exposure. And also, we are going to be giving the compound in an in vivo model to try to see if we are able to modulate the target the way that we like. Now, once this is done, and we have identified that, we move to the step where we're going to do a dose response. Basically, a dose response, it's going to be a relationship. You will have 4 doses, including vehicle, and you would like to see that the changes in the target is concentration dependent. Once that is going to be done, you're going to be doing a time course from where you're going to establish your target ED50 or ED80, and that is going to be the very first basis for starting to discriminate between your compound and your competitor. Now, once you are starting to get excited about a compound that you are feeling that there might be hope in terms of its potency relative to other competitors, what you're going to be doing is you're going to start giving this compound in a repeated dose, which is a multiple oral dose, at least one. In our case, sometimes we will do more than that just to make sure that we are better capturing the efficacy of the molecule and putting all of that in perspective. Now, once you are going to start the relationship and you have all this, so now the game is going to start moving from the biology to the Pkpd modeler or the translational scientist. What this person, he or she will do, they will start collecting plasma concentration and the PD. In this case, the PD, it's not just going to be efficacy, like, for example, like locomotor activities and stuff like that I mentioned. People are starting to put more emphasis on, and this is maybe like 15 years old, putting more emphasis on biomarkers and seeing how the drug concentration is influencing biomarkers. But we need to over here wait for a minute and ask a very important question. Why do we think we are putting a lot of emphasis on biomarker activity? It is very simple, because it is translatable. What does that mean? That means the target modulation that you are having in animal, you can also have typically the same modulation for the same target and the biomarker in human. So as a result, what can, now someone is going to ask me, like, who cares? I was like, no, you should really care, because what happens if you know the modulation of the biomarker over the course of the time, what will happen, you can use this biomarker in human to show that actually the proof of concept that your molecule does really work. Because if it doesn't, why should you move forward with the molecule? Cut your loss and move on with your life.
Ayman, 2 minutes, please.
Yeah. I'm getting there. No worry. Anything for you, sir.
Thank you.
A pleasure. So anyway, so now after we do all of this, what's going to happen after putting understanding the relationship with the biomarkers, the efficacy, this is when we start, putting the assessment of the potency of the molecule, what kind of a dose we should be targeting, and putting all of that in perspective. Then this is where the magic starts going to happen. After you put all of these important variables in perspective, you are going to start putting emphasis on the translation, which is what's going to happen in human. This is when we start taking our human dose projection and putting it in sheets to start making an assessment of what kind of a dose we are going to be having in human and what kind of uncertainty we should be expecting. Now, when you have all of that, of course, sometimes, and I have witnessed that, that when you are going to put, sometimes a molecule in human, sometimes there is going to be a sad story that it's not working. So this is going to be a great time for you to start asking why and to feed back to the drug discovery scientists so that they can identify a molecule and they can move on and identify a better candidate. So as a result, this is a round trip. And again, it re-emphasizes the importance of working in a team setting. Now, what's basically the take-home message is, number one, you need to have a good design and analysis interpretation of the studies in vivo. You need to involve early on the Pkpd modeling approach. The sooner you do that, the one who's benefiting is you. This is a teamwork effort, and also it will require feedback, which is, as we said, it's a round trip. Now, with that, I would like to have special acknowledgement for colleagues that I had in Pfizer, Jen Lin, Jeff Kaub, and of course, my current employer, IFM Therapeutics. I really have a good time there, and I'm looking forward to have a good discussions with you guys. Thank you.
Thank you very much, Ayman. That was a wonderful start to the session. I think you made the concept of Pkpd very lucid and easy to understand. I'll start off with a few questions that are already there in the chat box, and I request participants to ask their questions in the chat box so I can convey them to Ayman. So the first question that I have is, how have the expectations of the regulators changed over the last few years around the Pkpd data, which has been sought on applications, since all of this Pkpd concept has been evolving?
Okay. We need to emphasize, first, thank you very much for the question. We need to emphasize there has been a major change in how we are running our business, and also the acceptance of the agency for the data that's being generated from a Pkpd modelers. Now, you cannot submit any application without having this kind of relationship and these kind of descriptions. As a matter of fact, if you're going to be doing it the old school, I don't think they're going to be accepting it, or if they will, they're going to ask a lot of questions. Remember, the FDA is changing. First of all, they have inside the agency, they have a lot of postdocs who come in and work on these areas. So as a result, their knowledge of this area is really improving and at a very fast speed. There is also another concept that I wish that we can talk about, and it's very close to my heart. It is called PK, which stands for physiologically based pharmacokinetic modeling. Now it is almost rare, if not existing, that you cannot submit any application, especially for phase 2 and beyond, without having these kind of modelings, because they end up saving you a lot of money from a drug-drug interaction assessment standpoint and what have you. So So things are changing in a positive manner, and there is more emphasis on the use of these models. So that's why I keep telling people, especially the students, the young generation, guys, put a lot of emphasis on math and biology. The era of EC 50, EC Max is over. You need to integrate math in every part of the biology work.
Thanks, Simon. The next question I have is, do the Pkpd models cover the CYP profile also?
Okay. There are programs that I'm sure that you guys have heard about them, which is SEMCYP and GastroPlus. SEMCYP and GastroPlus, as a matter of fact, is improving, because SEMCYP and GastroPlus, initially they were placed to describe the PK of the molecule. Now they are starting to put more emphasis on the PD. So as a result, you can and you do integrate all of these things in a model. Are we completely there yet? The short answer is no, but are we working there toward that target fast? Make no doubt about it.
Wonderful. So the programs that you mentioned were SEMCYP and GastroPlus, right?
Yes, sir.
Okay. And these are these open source programs, or are they paid? So if students or any of the academicians want to access this, what's the easiest way to do this? Or are there any public open source programs available to do similar kind of Pkpd modeling?
Okay, good question. If you are a student or a faculty, you can reach out for Certera, the one that owns SEMCYP. And my understanding, they give you an open access with a minimum fee if there is a fee to start with for you to work on it. Okay. However, they have a condition that you attend one of their courses. And the good story with COVID, if you think about it in a positive way, there is now more and more online, so you don't need to travel, although you may like to travel. Okay. So that's one. GastroPlus also offers the same. But with that being said, there are also other PK models that are open access that people can use, and they are really worth getting yourself into it. If you can have your access to these, this is going to be they're going to be helpful. Me personally, SEMCYP, and I'm not exaggerating, changed my perspective about drug discovery and development. It helps me to integrate everything in one shot, and I'm a big fan. Also for GastroPlus.
Thank you, Ayman. Ayman, may I request that after your session, if you can just put this in the chat box, the information on these programs, and you mentioned Certera also. So if you can just type out the name of the company, that would be really helpful.
I'm at your service, sir. No worry.
Thank you. I have one more question. So
yes,
you showed data from animal model and correlating the PD with the pharmacokinetics in that particular animal model, right? Now,
yes.
is it sufficient to have a Pkpd correlation in a single species, or is it better to have multiple species Pkpd done in order for better prediction?
Okay. I don't like this question. I love this question, Dr. Narayanan. Okay. And if you allow me, I'm going to make a small tweak with your permission.
Please do.
It is not having it in more than one species. It is having it in one species, and it's going to be strengthened with translatable in vitro assays. You see the difference. Because why I'm saying this, sir, what happened, my friend, is sometimes you have species differences in terms of expressions of the target, in terms of efficacy, in let's say in mouse, rat, human. So as a result, if you're going just to put an emphasis on that, and you will, like for example, in IFM where I'm working on a target, and what happens is I have a mouse model. However, when I look at the efficacy of the molecule in a mouse model in vitro versus the efficacy of the model in human in vitro, I see that the mouse is 5 times more potent. So as a result, understanding this relationship, I should put this difference in my dose projection. So that's why I'm saying what you need to do is you need to move from the era of in vivo and put more emphasis on the in vitro assays to that basically mimic what happens in a human. Does it make sense?
Wonderful. I think that makes perfect sense.
Okay,
so,
sir.
Ayman, I think we've reached the end of the time for this session.
Thank you.
If there are more
Thank you.
questions, I request the participants to put them in the chat box, and Dr. Ayman will be available to answer some of them if possible. Thank you very much, Ayman, on behalf of DNDi, the Venture Center, and myself. Thank you for a wonderful talk. We look forward to a great interaction going forward.
Thank you so much, sir. And again, I would like just to end it quickly by saying thank you so much for honoring me and inviting me to this great session. I'm a big fan of what happens in India, and I think there's going to be great things happening in this amazing place. And you will see. It's a matter of time. You know, your guys are really very energetic, and I'm seeing, you know, you guys have amazing scientists. I have a lot of great Indian scientists who are really making a huge difference. And the big testimony of that, look who's now heading the Novartis. You guys have really great role models, and you should aspire to be one of them, if not better.
Thank you, Ayman. One thing
care,
I want to reiterate is we have not only an Indian audience, but we have participants from across the world who are joining into this session. So thank you for the kind words about India. I'm sure that this is the case internationally as well. Thank you once again for your session.
Take care. Okay.
Thank you. Having learned about the principles and modeling of Pkpd relationships, we now move to our second speaker, Dr. Anirudh Gautam, who has over 2 decades of pharmaceutical research and development experience in drug discovery research. Anirudh is currently the clinical liaison expert for branded markets and head of DMPK proprietary products at Dr. Reddy's Laboratories. Anirudh has worked on drug discovery research, clinical pharmacokinetics, differentiated formulations development, and also generics research in various therapeutics areas. He's an ardent believer of success in research. Alike, orchestra is an ultimate symphony of teamwork and collaboration. Integrator of product development strategies from concept to commercialization, he has identified and built alliances for new and emerging technologies, platforms, having possibilities of providing creative solutions and concepts for addressing challenging scientific problems. Anirudh's major research interests are in predictive modeling and simulation to support designing formulations targeted to a differentiated profile and establish translational models in early and late-stage product development. Without any further ado, I request Anirudh to speak to us on understanding Pkpd using preclinical models, lessons for efficacy studies. Anirudh, floor is yours.
Thank you very much, Dr. And for my kind introduction. And I'm honored for the organizers to giving me a chance to be part of this very important webinar series. It's very close to my heart. And before I start, let me just check that I'm able to present my screen well and everybody is able to see. Is this technology working right?
Yeah, Anirudh, we can see your screen. Request you to put it in screen show mode.
Yeah.
Thank you.
All right. So the scope of understanding Pkpd by using preclinical models is vast, I would say. And we just learned from Dr. Ayman how vast it is. And even the lessons learned are many and varied. So I would not be able to very comprehensively cover everything. But what I've tried in this presentation is that to give very, sort of practical aspects of this Pkpd modeling and usage, and in a very simple way, and hope people on this webinar would be benefited with that. with that, let's start, with my slides. And so the preclinical models. So let's understand what we mean by preclinical models, when we're talking about it. And to my consideration, we're talking about two things. We just heard, I may also tell that they are experimental models and there's a mathematical model part of it. So the experimental models can be in turn like acute disease models, where both PK and PD is studied in a single dose, one in dose level, sparse sampling, and monitoring single biomarkers. And then the subchronic or the chronic models will like next stage to that, where we study the repeat dosing at different dose levels. And here we start building the relationship between the effect and the concentration, or the drug response relationship. Coming to mathematical models, so what are these? These are basically, to me, an equational framework of the data what we generate from experimental models. Okay, so these require inputs from the models like in vitro EC50, in vivo exposure, in vitro studies, and then we get to what we just heard, that there's a direct mathematical model efficacy and the exposure and the indirect models. So as we see the direct models is that when we see the dose escalation, we see a dose response which is linear, and we clearly build a model. The left-hand panel in my slide shows this presentation, whereas but the life as we see is not that simple. Had it been so simple, then there would not been any struggle in this drug discovery research. But what we see that is an indirect model, the indirect response. That means there's a response is not matching point to point to the efficacy what we see or the PD parameters what we see, and not to but when more surprising is that there could be peak shifts, there could be change in baseline response, transduction, synergy. So there are many more things which come up when we study this exposure and response and try to build a mathematical model. However, the good hope is that the PK scientists with mathematical background, and I would not be shy to say that I include myself in that, have take this as a challenge and come up with more maybe complicated equations, but then they can describe the whole array of things which are happening, which is not very simplistic. However, the caution here is that we should still be going with the simplistic model, because the usage and the workability of the model depends on accuracy of the data what we're collecting from experiments and reproducibility of that. And that's why though they could be very complicated explanations, we have to see what is really reliable we can measure and quantitate and use it in the modeling. So right from preclinical studies, we should remember that and based further development based on that. So now let's start with what are key considerations for this Pkpd strategies and experiments. So the most important thing is that we should have the objective very clear. Okay, that means what we really want from this model, and then both experiment and the model what we want to build upon. Then we identify the strength, weaknesses, and gap, and what would be the possible outcomes from experiment. We have to think in advance and then have some assumptions built on, because this is like will guide us when we get the response, when we get the results, to sort of understand and interpret that, and then build on the mathematical models. Then, so we have to build assumptions. We will take care of that in our modeling as well. Then we have to select a sensitive and reproducible PD readout, and how we like the time effects on the PD outcome. So that is also to be taken into care. Then comes what we say that if we have some PK information, then we use PK efficacy data to guide the experimental design, so that the design is sound enough to answer the right questions what we have, to test the hypothesis we are building on. And so that is important. And most important is that when we are moving nowadays from the small molecules to biotherapeutics, we also have to take into consideration that the sometimes for the therapeutics, what we are increasingly seeing is that PK can depend on the PD by a process called target-mediated drug disposition, which means that the compound binds so efficiently with the targets and the receptors that it can impact the PD. So this understanding has to be kept into mind while designing experiments, and also same thing has to be carried forward when we design the modeling part of it. So next step, my next consideration is that ideally we should be using the same animals for the PK and PD readouts, but that is not always possible because of the animal limitations. And even sometimes the PK sampling can disturb the PD outcome. So we have to take into consideration. The solution is that we use satellite groups of animals, keeping in mind that the gender, species, strain, all the things are kept common, including the operator, to minimize the variability. Okay, so that's a very important things to taking care so that we are not getting something high variability which can impact. Then, even when sometimes these disease animals are limited, then we can go for the bridging PK. That means we do a PK in a separate set of animals which are of the same species, gender, but then they cannot be the disease model per se. They don't have disease animals. Yeah. So while I'm talking about all this, these have to be kept into mind, and the assumptions have to be laid out very clearly during designing experiments, and same thing has to be carried forward while doing the mathematical model of it. So one thing which is, I think, very much known and kept in consideration is that we have to keep for the PD part of it, we have to keep the vehicle treated groups as well, because the biomarkers may display the circadian variability, or it can in fact be affected by the formulations and all. So we have to minimize or negate that what we see that in vehicle treated groups versus the really treated groups. Moving on, the next and the very important part is the sampling. We should have a full sampling to describe the PK, to help us model, and then the PD also has to be there to have the modulation of PD is factored enough. So there has to be enough samples for that. And then comes to consideration the unbound plasma, T cell concentrations, and then the plasma protein binding. The species differences have to be accounted for real translation. So all these are the prerequisites and things to be kept in mind. So then with this experimental design, we can move to the mathematical model part of it, which is establishing the exposure response relationship and then simulating the exposure response relationship further from the next step. And for this translation, the in vitro potencies and in vivo plasma tissue levels has to be considered, as we've seen previously in Dr. Ayman's presentation. Then there are some PK considerations like distribution barrier between target and site measurements, and then all these things have to be made sure the PK binding are noted down, and then similarly for the PD, we have to see that non-specific binding pathways and all are considered, and they are carried forward, noted down and carried forward while building the mathematical model, so that the model is comprehensive enough and does a good prediction. Now, last and not the least, and Dr. Ayman also emphasized on it, and I'm also a strong believer that the partnership among the key or the complementary disciplines of scientists is a key to success for this whole exercise. And I will not restrict only to the scientists. The scientists have their own technical capabilities, but then talking and collaborating with the regulators, the internal regulatory team, and the agency we want to submit, and even the business and commercial people as important, because if their thought and their perspective also taken care, there could be some learnings which we are building in right in the in the preclinical stage, which can be useful, and they can be validated, and then acceptance of it can be taken care, especially by the regulators, and their utility by the commercial and the business is also taken into account. So, very much the success has to be, by a great collaboration. With that said, the question can arise that why PKPD has to be done at this stage, preclinical and early discovery development, when ultimately what will matter is the PKPD relationship in the patients. But briefly, what I want to say is that what we learn from here is very valuable going forward. So the effective design, analysis, and preparation of PK preclinical experiments can elucidate the possible relationship. Then it can explore the mechanism of action, and then we can identify the PK properties which are actually guiding, the PD, and that can be used to optimize the compound for the discovery and formulation for, for the delivery type of, interventions or research we're doing. Coming to modeling, Pkpd modeling, the increased translation of in vitro potency to in vivo setting is understood at this stage. Then these studies help in reducing the number of in vivo studies which we can perform. Then it helps in improving translation of preclinical species to clinical setting and shorten the development time. And this is a chance where we build the therapeutic index of a compound or a formulation to limit ourselves or to guide the clinical development part of the drug development. So here the emphasis is actually in selecting the compound or formulation to identify the potential safe and effective usage and the dosage arrangement as we move forward. All right. So then, with this very comprehensive, then it should look that, we have covered everything, but then we should always remember that there are some limitations to this Pkpd strategies. And just to highlight a few of them, the first one is the variability. The variability is a big killer, I would say. It can make or break the experiment and the whole hypothesis. So there are sources of variability are many. We can't control everything, but then all those has to be at least accounted for and listed down right when we're planning these experiments. Then there could be that we talked about the biomarkers. Then sometimes what we not have is a valid or robust or clinically validated biomarker. Even if it is there, then its amenability to be using for continuous sampling and modeling simulation may not be there. But so in those cases, what we do is that we can use we can start with a tool or a reference compound to build this Pkpd models, and so that we validate that before we actually do our own data analysis and experimentation into that model. So that's a strategy to be taken. Then comes another challenge is that sometimes what we have the PD readouts as very categorical or binary responses like effect, no effect, death or survival. So there's no continuous thing. So then how do we deal with this? Is that we have to have the adequate exposure, found out, and this could be done by dose fractionation and order dosing regimens to elucidate the dynamics, the cutoff points where we are getting into, the two binary outcomes. And as I said, the mathematicians and the statisticians have their own way of putting the models. There are different models to that, like the proportional odd models and logistic models. So with this data in hand, these sort of categorical outcomes can also be modeled, not just the continuous model where we can have, as I described previously. So these are some limitations which we have to keep in mind while working at the preclinical level itself. And then this can be taken forward at the clinical level as well for the Pkpd. Okay, so with all this understanding, the actual value of this, all the preclinical Pkpd experimentation lies in translation or translating it to the clinical setting. And this slide gives us a simplistic framework of how we do it. So at the preclinical level, we identify the PK indices, as we call, which are responsible or correlate with PD indices, respectively for the efficacy. And then we have to adjust for the difference in target pharmacology from the animals to the humans. And in parallel, that's for the PD. And for the PK, we have to scale the PK to the human, and we do that elementarily. They are well-established, theories for that. It can be done. And then we simulate the human PK, and we put the dose and dosing regimen, which is then used as for the next level of clinical development stage, right from the first human studies and all. And as again, Dr. Ayman has mentioned, that the feedback, what we learn from PK is also important from from animal to human, we have to get the feedback from the PK and PD, which is not just used to rework, but also to refine the Pkpd model and extending it for designing the very late stage clinical development as well. Okay, so this is the whole framework we have to follow and kept it in mind. Now the good news is that all these efforts, and there was one question, and this, I'm trying to address that indirectly over here, that this model-informed development, right from the preclinical stage, and the totality of experiment of the evidence is looked by the agencies as a very important support to make the decisions and maybe assign or get the confidence for the approvals. So this whole data is being very extensively encouraged to be developed and used for the entire process of discovery and development. Moving on, before I get to some case studies, I would just like to sort of give a glimpse of the span of preclinical Pkpd strategies and their impact, which is possible. We know for new chemical entities, the discovery and development, it is a very stepwise characterization and linking the individual potency, then the target exposure, engagement with the pharmacological activity, mode of action. We just saw that, and then efficacy which translated into humans. So that's very well understood. However, for the new therapeutic entities development as well, which is basically like repurposing of the molecules, we have seen a lot of that type of research happening in the industry right now in many different ways. And then the differentiated formulation development, and then the delivery systems, all those also very much use this preclinical Pkpd strategies. And here what we do is reverse translation, which means that we already know about the compound and maybe or the clinical stage, and we hypothesize for a different purpose or benefit in the formulation development or differentiation, and then we design the experiments in the preclinical level again, test the hypothesis, and then move on. So that's how the whole entity development happens. By this way, we can cut down many of the experiments, and we can just go ahead and submit a very scientific rationale for not doing very extensively, or maybe doing smaller clinical developments at a later stage for this type of approaches. Third, and what I've captured is, and this is growing, the usage is in the biopharmaceutics and the formulation development strategies. Most important is what also Dr. Ayman was mentioning is PK modeling. The physiological-based PK modeling has been very much extensively used. There are guidelines coming up, and in fact requirements coming up by especially FDA and EMA, where we are supposed to have this PKPD modeling done, which helps in prediction of absorption prior to first in human studies, then formulation development and dissolution profile development, and optimization of the methods as we move forward in the clinical development. Then also the modified release formulations. As we know nowadays, it's not the simple IR formulation might be useful. They could modification has to be done in an appropriate formulation development. All these ways, this modeling is being used to guide the decision making and progression. And sometimes the late stage formulation could be very different from what we started doing in the clinical development is first at the early stage, and that bridging, the bioequivalence itself can be predicted and sometimes used to get biowaivers, especially for the what we call as the predictions of PK in the fat studies for the BCS class 1 molecule. So this is very much happening and being used. The role of Pkpd is expanding more and more, and at the preclinical stage itself. That's what I'm trying to say. And the reason behind that is that the quantitative information is they're obtained in this preclinical Pkpd relationships.
Anirudh, you have 5 minutes.
Oh, okay. So I had taken 2 case studies. Maybe I would do only one. Let me see. So the first one is very recent, and from the molecular development for the COVID, and and why I took this example is that we've all been watching, and there has been a lot of publications and very interesting stories to tell of use of very specific models for this development was taken care. to not waste time getting directly into this, what beside the usual models, what the developers has used for this is a human lung only model, where it is basically an immunodeficient implanted with authentic human tissue lung. Human lung tissue, basically. And what this helps is it gives a one platform to cover all the SARS and MERS viruses. And then here, the replication, the thing is that replication happens in this model without extensive adaptation of the virus per se and the genetic adaptation for the host molecule, because the host is basically here in the mice, and otherwise it would not be possible. And so that is the the beauty of this model, and this was used, and what we was seen by Molnupiravir was that once if the treatment is started maybe 24 hours after the drug exposure, there was a 4.4 log decrease in the titer of the virus. But if it started after 48 hours, the titer was decreasing, but it was just 1.5 log decrease, which is like 90% decrease. So what it reveals is that the timing of dosing is very important. Initiation of drug treatment is very important for an antiviral to work on this, this situation. They also tested the prophylactic uses of it by a very specific, treatment starting 12 hours before the induction of the SARS. And what they saw is 100-fold, exorbitant, decrease in the titer. So this was a good learning which was taken forward in the development. And the second model I wanted to bring in is the ferret model, which was used. This model has a thing which is specifically being used by for this development was that the transmission, how to get the information of transmission control of the treatment. And this model, this ferret has can transmit the virus efficiently with minimal clinical signs, which resembles the spread which was happening at the stage when this Molnupiravir was being developed. And so it was validated model. And then what they did is the picture on the top shows the design. I will not go into details, but what they saw was that again with the BI, the morning and evening dosing of two regimens, two dose levels, they saw a significant decrease in the viral overload in the upper respiratory tract of the animals, and it was also time dependent. So the graph, the lower first graph shows of that if the red line, which is shifted outwards, that it started later on 36 hours after the treatment of the infection, then the response is delayed, and the black line shows the placebo when there is no treatment happening. So clearly a time dependent is important for the treatment initiation. And the other two graphs on the right hand side talk about after there was complete silencing of the virus. There was no replication going forward again. So this was also very important learning in the ferret model, which was taken forward in the experiments which was in clinical trials. And just to cover one more is that even the contact spread was tested in the same model, very diligently devised experiment, where what we saw is that when the animals which are housed with the ferrets which are treated, then there is a clear-cut suppression of the untreated. The untreated animals don't get the COVID, the virus into them, and so this identified that this is a promising antiviral countermeasure to break the spread of the transmission chain of the COVID. This was further taking into account this dynamic in the mathematical modeling and what is called as dynamic viral model. And this graph shows that the viral modeling described is that the black one is a wild type model, wild type virus model. The days the viral load shoots up and comes down, and the other on the left hand side what we see are the other viruses where they have a different degree of transmission, and the red one, which is the R0 of almost like 2, shoots up faster and then comes down faster as well. And what I was seeing in this experiment of modeling is that with the therapeutic effect, a therapeutically treated course, the total viral load, which is measured as an AUC, is significantly up to 70% decrease for the most variants of concern, which is high transmitting variants of Delta and Omicron, which is shown in the red curve, the almost 70% decrease. And even the permanence time is also significantly goes down, especially for the high Delta variant. So this again was used as a it has a role, the molnupiravir has a role in the pre and the post exposure prophylactic use of antiviral treatment for the variants of
Can we conclude in a minute, please?
Yeah. So that was my first case study. Second was I was talking about how is an in-house experiment, how we used to use this preclinical Pkpd experiments to decide upon the go-no-go decision. And I'll not go into details, but what I would just show is that we systematically did the in vitro experiment and ex vivo experiments where we tested two molecules and we got very positive results initially, both with the efficacy and potency and the rat exposure, where we thought that we have at least one selection to be taken forward in clinical. But when we went into in vivo study, in the constitution which is used, what we realized that the target which was there to have a local action was not achieved by any of the molecules, and then a decision of dropping this for further clinical development has to be taken. So in the industry, this is also very important that if we are able to decide upon a fast kill, this helps a lot of efforts and money to be saved to not taking molecules which may have some positive results upfront initially, but may lack or may or see a major challenge later on in the clinical development. So that's all. Finally, there are three future trends which I was thinking that to share. What I'm seeing is this, all my perception, it's not that it is very much there, but then I see that very much coming, is that we would be moving from the culture organs to disease models which are cultured, not using ethically the animals which we're using until now. Then the AI will also have a very positive impact, and we'll be able to link multiple Pkpd models, especially for the combination drugs. And the third one, which again very much talked about, is that the blockchain will enable data sharing, and there would be one day we will be doing open innovation, at least in preclinical setting and translation to the actual clinical setting. So these things I think are coming, and here the preclinical Pkpd and the Pkpd modeling would be playing a greater role in moving forward. So thank you. So though it was fast, and I hope it was helpful to the audience over here.
Thank you, Anirudh. I think that was a very informative talk on how preclinical models could actually be used in Pkpd modeling. I have a few questions from the audience, which I would bring up. So while the Pkpd modeling for the oral route is fairly straightforward, what are the key factors to be considered for topical and inhalation formulation with respect to Pkpd modeling?
Yeah, so very good question, and I think this is what is coming up, because obviously oral, though more preferred, is not always the viable or maybe differentiating route of administration. And so yes, they are specifically laid out for the transdermal or dermal thing. What we have is that we have to consider what all parameters, and where we go to IVPT studies, which are instead of visual studies to be taken care. And then we have models, both ex vivo models of tissue, where we see the absorption and the movement of the drug from the formulations to the tissue, the dermal level, and depending on whether the indication is for the just for topical on-site application, or maybe going into the system. They are established models which are coming up, and And in fact, the GastroPlus is also coming up with very defined modeling tools, and we have the defined parameters which can be studied in experiments and then built into for these models for both dermal, nasal, and many different routes of administration. So yes, there is a possibilities. The design has to be very specific, to the intent of administration.
Thanks, Anirudh. There's a second question on how PKPD modeling will help in the development of novel drug delivery systems.
Yes, as I mentioned, so the most important thing is that when we know the target of the novel drug delivery system, we have to design experiments, and the modeling can be done, which shows that what could be target PK profile, in the delivery system being obtained, and depending on what route of delivery system it is, and and the formulation type, there are particular aspects to be inbuilt into both the experimental model as well as mathematical model to take care of what we say as the target profile of the formulation, and then it has to be tested and validated, and improvement that can be done with the feedback mechanism which we talked about before we go to humans. And then ultimately, once we have a shortlisted drug delivery system which meets our target hypothesis of formulation as well as the target PK, and the PK is obviously linked with the PD, which would have been either developed by the known route of administration of oral or intravenous, and then this bridge, the gap can be bridged and then and tested in humans in healthy volunteers to get the validation or improvement required per se for this. And this has been, we've been fortunate enough to be in this, myself being into this role for helping the formulation in the differentiated delivery systems to be built up, and we have some good successes with the getting in the US it's called 545B route, where the delivery systems are very different, and use of this PKPD modeling has helped us achieve those successes.
I have time for one last question. The question is, what is the appropriate model or strategy for cross-species PKPD?
Maybe I'm not able to clearly understand the question. Like when we say about cross-species, obviously the difference in the the pharmacological aspect and the physiology of the species has to take care, but the data generated in different species is ultimately used, and with with those considerations taken into the PKPD model, is what we can build a robust model and use it for the further translation to humans, if that's a question is being
I think that's what was the question, and I think your example of molnupiravir actually addressed that to some extent in terms of the PKPD modeling across species and the importance of that for predicting the human dose as well. Anirudh, I think we are out of time. There probably are one or two more questions in the chat box if you don't mind answering them. Thank you very much again for a very informative talk, and on behalf of DNDi Venture Center and myself, thank you very much once again.
Thank you for giving me the opportunity, and thanks to the audience for patient listeners.
Thank you.
Thank you.
Now, moving on from PKPD, I think we move to another important topic in drug development, and that is on the safety aspects of a molecule during the discovery and development phase. And to talk to us about the role of safety pharmacology, interpretation, and translatability in the clinic and first in human studies, we have none other than a very eminent personality, Dr. K.S. Rao. Dr. K.S. Rao is a MBSc, PhD, and a board-approved, board-certified toxicologist with more than 40 plus years of global experience in safety evaluation. He started his career in 1971 at GD Searle, and later moved to Dow Chemical and Quintiles, USA. After returning to India, he has been providing guidance on product development to hundreds of companies across the world, with major emphasis on testing schemes, strategies, and risk assessment. He's an emeritus member of the Society of Toxicology in the USA. Dr. Rao is a recipient of the Toxicologist Mentor Award from the Society of Toxicology and is the first person of Indian origin to receive such an award in the 60-year history of its existence of SOT. Dr. Rao, we are really fortunate to have you as part of the speaker panel. Over to you, sir.
Thank you, Dr. Sridhar. I appreciate the introduction. Let me start my presentation. I don't know, that started my edit.
We don't see your screen, sir.
Okay, one second, let me just. I just clicked on the share screen.
You will probably need to select the window which you want to share.
Oh, I see. Yeah, that's right. That's yeah. Is it okay now or not yet?
Not yet, sir.
Dr. Roy, need be, I can share.
Yeah, please, why don't you please go ahead? I think there is some issue from my side. First, I want to start with a little apology. I live outside of Bangalore near the airport. The internet system is not that good, so I'm on a hotspot here. Please bear with me.
No problem, Dr. Rao.
Can you hear my voice okay?
Absolutely fine, sir.
Okay, good. Okay, we'll start with again, the safety. The subject was given to me as safety pharmacology, which actually in my mind belongs to the previous session of toxicology, but still I will make an effort to sort of make sure the right audience is here for this presentation. Next slide, please. Okay, first let us have some definitions of pharmacology and toxicology, which is absolutely very basic. The reason I put is there probably are some graduate students who probably needs a clear understanding and definition of this. Pharmacology, all of you know, I don't have to define therapeutic effect. The toxicology is the one where I want to emphasize. The toxicology, the study of adverse effects on xenobiotic agents. Again, we're talking of the adverse or harmful effects, or the effects beyond the therapeutic effects. So those are adverse reactions from drugs. Just to give an example here, I have given morphine as a CNS, which is a respiratory depressant, which all of you know. Whereas example is acetaminophen, which very few would know. They take it every day, probably more often. It long-term usage causes liver damage. So that's actually an example of toxicity. Next slide, please. Okay, now we'll come to the proper subject of safety pharmacology. I do not expect a fair number of people may not have had too much exposure to the safety pharmacology. Typically, safety pharmacology studies can be phased in the discovery phase, in the screening modality. But I'll come back to the history later on. But by and large, these safety pharmacology studies, the candidate has been lead identification has been done. So safety pharmacology is simply a branch of pharmacology specializing in the detection of investigating potential undesirable pharmacologic effects, which will come back to exposures in the therapeutic range. The key thing is in the therapeutic range, we find some abnormal effects, which has been observed in a number of drugs after they are marketed, which we'll discuss later on. And these safety pharmacology studies have guidelines which have been published by the International Conference of Harmonization and plus various other governments also, ICS-7A. It is a very evolving field. It is one of the latest field in the area of safety assessment. It is a combination of physiology, pharmacology, and toxicology. It's a kind of a blending of all these three disciplines. Next slide, please. Okay, now here I'm just giving, I'm not going to read the entire table. My slides will be available through DNDi on the website. You can view it. So the just major points I want to touch here. Toxicology generally applies to the repeated dose. Very rarely we find one single dose will produce toxicity. That very rarely happens. But safety pharmacology, usually you want to find a single modality, a single dose at a therapeutic or a slightly higher concentrations of the plasma concentration, which Eamon presented in the first time. The major endpoints in toxicology, number 2 point, is structural changes. In addition to the food body weight and clinical chemistry, the predominant effect in toxicology is the most sensitive one is structural. Whereas in the safety pharmacology, endpoints are usually very short-term. By and large, many of them are reversible, and they are functional effects recorded from conscious animals, usually. And we'll come back to the animal models in the next series of slides. So in the area of toxicology, as I alluded to, organ weight, hematology, those are the classic parameters, including histopathology is observed. Whereas in the area of pharmacology, we evaluate the onset, duration, and magnitude of the effect. That's very, very critical, which is not commonly done in toxicology, because toxicology, by and large, they're very terminal. One of the last one, which is dear and near to my heart, is the area under the plasma concentration, which Eamon alluded to. I want to re-emphasize that is absolutely very, very critical, not only in pharmacology, but also equally important in toxicology and safety pharmacology. The plasma concentration and the area and the systemic exposure, AUC, is very critical. Whereas in the safety pharmacology, is predominantly driven by the Cmax. The Cmax is the one which determines, that's where the pharmacologic effects are observed, and therapeutic effects, that's where we look for. Next slide, please. Okay, now we'll ask about what are the different models. I'm not good. There are literally dozens of models. So obviously it is very difficult in 30 minutes to describe all the models. I'll classify them into 3 major categories. Like pharmacologists, many of you know, there are in silico models available, prediction models. In vitro, wonderful models are available, Herd model. And predominantly, most of us depend on our in vivo models, which can be translated from animals to humans. That's the very basis of drug development. Next slide, please. Okay, what are the organ systems? Again, there are 3, which is no news to all of you, 3 major what I call vital organs in the body. One is the central nervous system. If you don't have, obviously, that's end of the individual. Cardiovascular, similar thing, and respiratory. These are the 3 vital organs, and that's why the safety pharmacology is centered around these 3 vital organs. However, after these, in special cases, if certain drugs has certain secondary effects, we also look for secondary organs, for example, GI tract and the renal system. Next slide, please. Okay, what are the key aims of safety pharmacology? Is to protect phase 1 clinical trials, because we are administering our potential NCEs, new chemical entities, to normal, healthy individuals like you and me, most of us. We do not want to harm them. So essentially, we want to protect the phase 1 volunteers from acute adverse effects. That's why all safety pharmacology studies, by and large, are tested in a single dose modality at a Cmax concentration. Now, second one is to protect patients. Next one, the phase 2, obviously, we may have a therapeutic effect, but there are, if the adverse effects related to the drug occur, which becomes very critical for survival of the patient, that indeed has happened in a number of drugs after the marketing. This is unusual. So we want to protect the phase 2 patients, patients. And also, finally, to minimize the risks of failure during drug development, and more important, post-marketing. That is the worst kind of a drug episode we would like to have, but unfortunately, that has happened in previous cases. Next slide, please. Okay, some can be phased in discovery, as I alluded to. And these days, most of the big pharma companies, the top 10, they do that routinely. But however, by and large, bulk of the so-called SMEs, small and medium-sized companies or virtual companies, Many of them I deal with through my CROs. They don't have any idea of these things. They come up with the one or two lead molecules and then say, let's do the GLP tox. That's how they want to get into the phase 1. That is the wrong attitude. If there is an opportunity and time, or make some time for it, do some screening for safety pharmacology very early in the game. But these are usually conducted once the lead candidate is identified under good laboratory practice. Most of you know it. I don't want to embellish on that. And these are conducted usually prior to, and in fact, always prior to the FIH, first in human administration. However, after having done that, we still follow the same parameters in clinical phase 1 to phase 3 all the way through. We want to make sure nothing is left to chance there. And before approval, obviously, NDA application, there are some investigations can be done based on what we find in clinical. So this is a continuous process of preclinical assessment based on the pharmacology data, based on clinical phase 1, 2, and 3 data. And even some post-approval safety pharmacology studies have been done. Next slide, please. Okay, now safety pharmacology studies I alluded to earlier, the guidelines. These are the two major guidelines, S7A ICH, International Conference of Formulation, A and B, and all safety pharmacology studies must be conducted according with these guidelines and under GLP. Next slide, please. Okay, now question is, what are some of these factors to consider? Again, these are a lot of them. I don't have probably time to go in depth. The slides will be available. What to test? The test systems are very, very critical in these things. Again, this is not a generic test. You just simply take a rat or a dog and do it. No, you need to look at the pharmacology, the class effect, the type of molecule that we have, structure, activity, all those things need to be considered. Then in vitro, ex vivo, cells, membranes, tissues, organs, all these things come into the picture. Even though it looks very simple, but it's extremely very complex. Test item formulation, very important. A lot of the people have difficulty in what to test, which product to test. And then the route of administration, obviously, it has to be similar to the human. Dose levels, unlike some of the other tests that we have been discussing before, the dose levels and test concentrations are very, very critical. You don't want to harm the animals where it becomes sick. You want to test at a dose which is slightly above the Cmax or around the Cmax, but not too far beyond. And define the concentration relationship, very critical in all drug development. Next slide, please. Okay, now, as I think I already mentioned, discovered dose level, safety pharmacology, dose level in all the pharmacology and toxicology, dose response is critical, which I don't want to spend more time on this. Next slide, please. Okay, now CNS. For now, we'll go to I alluded earlier, there are 3 vital organs we're looking for: CNS, cardiovascular, and respiratory. We'll take the CNS first. Critical for drugs intended to act on the brain in the CNS central cerebellum and spinal cord. The second, obviously, critical for drugs with an ability to cross the blood-brain barrier, BBB, which is again a common sense here, and critical for drugs known to interfere with the CNS functions. These are the 3 criteria. Next slide, please. Okay, what do we do in these things? Again, this is a study of a study outline, or what I call a high-level design of a study. Overview is a global neurological behavioral functions we study. Typically, they're done in usually rats or mice. Typically, I would prefer a 6 per sex. Some people go only male or female, either way, but prefer to include both males and females here. Obviously, in all toxicology or pharmacology studies, dose response is very critical. Control and typically 3 dose levels. Where you are very sure the dose levels are tolerated, we probably can get by with low and high dose also. Intended route, obviously, it has to be similar to clinical, but in some cases, I'll give an example. If the exposure is dermal, where the absorption is relatively low in animals, then in that case, I recommend doing intravenous safety pharmacology studies based on a case-by-case basis. Typically, what we do is when we administer the drug, we observe. FOB stands for functional observation battery at periodic intervals up to 24, even beyond some cases to 48 hours. That typically after giving the drug at various these time points, we measure approximately 30 different parameters. All these things are listed here. Next slide, please. Okay, this is just the next one. We talked about the CNS first, then we'll go to the respiratory. Typically, respiratory safety pharmacology is done most often in rats because most of the equipment is designed, and secondly, we have a lot of toxicologic data and PK data in rats. Just like the previous slide I mentioned, is very common and must be done control in 3 dose levels, dose response, route similar to humans. What are the parameters we measure? On the right side, you see the pictures of the rats sitting in a cage. Usually the drug is given, then it is transferred to the plethysmography for monitoring the respiratory parameters. The parameters are respiratory rate, minute volume, and tidal volume. These are the absolute basic. But based on your class effect and the type of a drug and a variety of other things, we need to consider other parameters on a case-by-case basis. Next slide, please. This is just a pictorial description or the showing of our cardiovascular telemetry. Before we come to the picture, we can be watching that picture. Typically, cardiovascular telemetry can be done in either in dogs, monkeys, or even sometimes pigs also. Typically, what we do is we actually tie a belt around the animal where actually remote electrodes are inserted, typically subcutaneously. But these days, some jackets are available in the Western world. Unfortunately, they're not available in India, because I happened to sort of ask one of the supplier at the Society of Toxicology meeting. The guy was so careful. No, we won't give it. You may copy and duplicate this. So essentially, they're very protective of that. So as a result, these jackets have not come to India so far. So typically, we insert the subcutaneous electrodes. Next slide, please. Okay, typically what we do is cardiovascular parameters are done only in free-moving conscious animals. So I want to make a case here. The bulk of the facilities that I worked in the US, they are all so carefully constructed, where absolutely not even a pin drop silence. If a pin drops on the floor, the dog will not hear any song. That kind of a silent modality is typically done, and that should be the modus operandi. But unfortunately, there is not a facility that I have seen in India which has that type of facility. Still, we do this, but somehow I'm not quite happy with the cardiovascular telemetry systems in India at this time. Hopefully, people will improve over time. Typically, as I said, they are done in dogs and monkeys. Typically, we do a crossover study using 4 dogs. Those of you who may not be familiar with what a crossover is, typically what we do is we take 4 dogs. First is given the control, there's the placebo. The second is low dose, mid dose, high dose. Then we rotate all the 4 dogs for all 4 groups. In that way, we have the same animal going through all doses. So we get a very good comparative data on the same animal, different doses, including the placebo. So intended obviously clinical route, again, dose response, and continuous monitoring. Once the drug is given, actually the animals will be floating around with this implanted electrodes remotely through a glass window. We usually monitor their ECGs and blood pressure and other things, heart rate that are indicated here. The critical points, this is the most important thing, very few sort of adhere to, is correction for the influence of heart rate and QT. Because many pharmacologic compounds, you are all pharmacologists, you know, compounds have a tendency to increase heart rate and or blood pressure. So we need to correct, particularly heart rate. Heart rate has a big impact on the QT interval. So we need to correct for the heart rate, particularly if it increases more than 10 to 15%. The adequate validation is very important. It is not enough to simply implant and say, hey, I've done 1 study and reason. Usually I would not give this study to any CRO which has done less than 10 studies. So that's again, I'm being tough here. So we need the good historical control data. So adequate and also diurnal variation. We need to find out a lot of it, as both Eamon and Anirudh mentioned, variability is very, very common in these things. We are using only single dogs here per animals and calculating this and extrapolating to 7 billion human beings ultimately. So essentially the variation is very much. So we need a good cardiovascular pharmacologist and also preferably a veterinary pharmacologist who can actually analyze the data. In fact, there is none in India veterinary cardiovascular pharmacologist, and we need to improve the technology and training programs in this. I'm emphasizing this. So, and also historical control and autonomic influence on the animal. Next slide, please. Okay, dog telemetry typically is data from a free. Next one, sorry. Okay. Key issues to consider in safety pharmacology in general. So again, I was hypercritical on the dog telemetry because that is the single most important study that we do among these 3 safety pharmacology, which is far-reaching importance and a kind of almost devastating influence on the several marketed drugs, which I'll come in the last slide or 2. So the detection of adverse effects and liability, this is very important. Key issues to consider: investigation of the mechanism, which Eamon alluded to, very important. Mitigation strategy. Already you do find something. How can we manage risk assessment and risk mitigation is very critical. Not just enough to find an effect, how to mitigate that. And also calculate the projected safety margin. As both of my previous speakers indicated, animal studies by and large are okay, but they're not definitive. So we need to actually project and predict what could theoretically happen. At the same time, always err on the safe side of safety. And also consider everything we do in toxicology or safety pharmacology, consider always what is the implication for clinical monitoring, impact on clinicians, so that convey the data in a succinct way, summary, succinct to the clinician as to what he or she can expect in the phase 1, particularly phase 1 study. Next slide, please. Okay, now supplemental studies I answered. Usually, I would say 85% of these drugs that are developed, they routinely go through only the CNS, respiratory and telemetry studies, and HERG, which I didn't discuss. The supplementary studies are a case by case. They are done urinary, autonomic, and GI tract. They are probably done about 10 to 15% of the time. Next slide, please.
Okay. Dr. Rao, you have 5 more minutes.
Yeah, I'm completing. Thank you, sir. Okay, good laboratory practice. I already told in my first slide, all these studies after the lead identification must be done under GLP. Next slide, please. Okay, the story of terpheridine. This is one of my favorite drugs. This is probably the classic example I want to give. This changed the whole science of drug development, and this actually created the science of safety pharmacology. Terpheridine was a drug marketed by Novartis back in 1985. It's the first non-sedating, is almost a miracle antihistaminic drug. Terpheridine acts as an antagonist to a histamine H1 receptor. In those days, other receptors were not known. It is a prodrug. It is when you give this drug, or when they gave the drug, they could not find hardly any of the parent drug in the plasma compartment. Then later on, they discovered it's totally metabolized by the CYP450 3A4 in the humans. So due to your near complete metabolism by the liver, immediately there is nothing left in the plasma compartment. So the example that actually precipitated is ketoconazole, which alters the metabolism of CYP450, which indirectly affects the terpheridine, resulted in the accumulation of unmetabolized drug. Just to make it, let me repeat this. This is the single most important point I want to make before I complete. As when a person gives get both drugs, ketoconazole, after the terpheridine is given, so you see a significant marked increase in the terpheridine concentration because CYPs are inhibited by ketoconazole. And that was not apparent in the toxicology studies prior to. They've done every study conceivable. Novartis is the best company. They have done everything they could, but they didn't know the safety pharmacology and drug interaction, which Eamon and also Dr. Sridhar alluded to. So following marketing, they found a very long QT prolongation and almost near lethality that happened. Immediately, the drug was withdrawn, or in fact, the manufacturer himself decided to withdraw. Obviously, they don't want to take the risk, and finally withdrawn. Then the fortunate thing in this case is a last comment I want to make. The Novartis was extremely lucky. The metabolite of the terfenadine, fexofenadine, is a drug and is subsequently marketed that does not have the drug interaction. So that's kind of a success story there, with starting with a bad episode. Next slide, please. Okay, this is just the same thing I talked about. I don't want to spend too much time. In a nutshell, I summarized the whole thing in one slide. I think that this stage, I want to stop at this stage, and I want to give a chance for people to ask questions. So this is one. There are many. There are nearly over a dozen drugs are withdrawn because of this, because of the QT prolongation, not one or from development. So this is a very significant one. So essentially, safety pharmacology becomes very almost, in many cases, a go-no-go decision. Thank you very much. And I want to open up questions. Dr. Sridhar, it's yours.
Thank you, Dr. Rao, for a very informative talk. I have a couple of questions from the audience. So the first question I have is, what is your expectation from the US FDA under the new approach method strategy of implementation of safety models, especially case of CNS evaluation of NCEs?
CNS evaluation, as I indicated, if a compound goes through the blood-brain barrier or the CNS spectrum, but by and large, any small molecule, any small molecule, I didn't touch one more thing, which I'll talk in a minute. All small molecules, particularly comes to the for the IND, must go through these three things, unless there is a special reason. Talking of this large molecule, which I did not touch, most of the large molecules, they have a specific target. Then most of the large molecules do not have an off target. Small molecules, usually the biggest disadvantage, and that's why we have adverse effects, is in addition to the very high efficacy, they have numerous bizarre non-target effects and also high variability among populations. So as a result, for all small molecules, by and large, I would say more than 90% of them, these testings are absolutely mandatory. For large molecules, certainly our peptides and proteins, we certainly don't need this, not required. However, by and large, what happens is many of the larger companies, the cost of the safety pharmacology studies is approximately 100,000 plus. When you are already spending a couple of $1 million only in preclinical, so let's do this. So generally, by very many large companies, they do these things as a kind of a routine practice.
Thank you, sir. One other question I have is, how reliable are zebrafish models for predicting cardiovascular toxicity? And
sorry, I missed the first half. Can you repeat the question once more?
How reliable are the zebrafish models for predicting cardiovascular toxicity?
You know, that's one thing just came in the last maybe 5 years. It's coming up, and I have not worked myself with zebrafish, and I have not really done a thorough literature search on that. However, one thing I must tell you, Dr. Sridhar, and also the audience, the zebrafish is predominantly used in the screening modality. So it has not come to the stage of where it has come to a guideline study for GLP studies. As a result, probably those who are in the discovery mode, particularly the top 10, top 5 or top 10 companies, I heard they are doing in the discovery mode before the lead identification is done. And that data is not much available, publicly available. As a result, I cannot comment on the predictability, but it's not routinely, but fairly commonly used by a number of big companies in the screening mode.
So I want to just add to that on my own as well. In terms of looking at some of the safety aspects, are there in vitro models which would potentially be used and approved by the US FDA as studies which could be part of your regulatory package?
Well, bulk of the in vitro models are actually buried inside the big companies. They do these things. And unfortunately, they are not published much. But the few publications that I have seen coming out from the universities, very good universities, they are really not come to the stage of maturation where the regulators have accepted. As a result, at this stage, by and large, for regulatory purposes or IND purposes, we still depend on these three in vivo models. So in vitro models, predictability, since my knowledge is limited there, so I cannot comment on the predictability or what is the likelihood the in vitro will become officially reality. But the cost of these, the key thing is the cost. The cost factor of these, all three combined together, we are talking, even in the US, we are talking a total $150,000. So as a result, the cost is not an inhibiting factor for developing, doing these studies on a routine basis. The in vitro models, the zebra model, all these are absolutely very important in the discovery stage, where you have, for example, 3 or 4 molecules which have a similar therapeutic ratio or activity or things. You want to pick up one. In that case, certainly it can be used there. But when it comes to the lead or post-lead identification, I would not depend on any of the in vitro when you have the in vivo studies at a relatively less expensive cost compared to the totality of the expense that is involved in the just preclinical itself. Millions of dollars are spent.
So one more question is, how do you decide that the dog is the best non-rodent species for your molecule?
I missed the first part. Can you repeat once more, please?
Yeah. The question is, how do you decide that the dog is the best species for your non-rodent, for your molecule?
Sorry, I didn't hear the first word. Okay, I got it. So I know that is a $64,000 question that there's no easy answer. By and large, again, if somebody would have asked me 50 years, I would have said, I don't know. But today we have so much of historical control data on hundreds and if not thousands of drugs that have come on the market. And we have a lot of both published information and also experimental information available. So we are so confident that unless you have a specific target specifically present only in the primates, so dog has become almost a universal non-rodent model after the rodent. So essentially for that reason, the regulators have recommended. And also one more publication which I mentioned from this was back in the 70s, if I remember, late 70s, a publication came out from the industry where they actually compiled data from a large number of pharmaceutical compounds, both under development and marketing. They actually compared that if they for 60% predictability, if they do only dog studies, maybe another 55 to 70%. So when they do both rat and the dog combined, they found that the predictability goes up to close to 80%, if not quite there. So essentially, again, with the 80%, that still we have a 20% probability that drugs will fail, no question at all. So that's based on that. That's what rat and dog have become almost the default species in drug development. However, in the case of a large molecules or in small molecules where there's a special affinity, a receptor, or the target organ present in either the primates or in mini pigs or any other species, then certainly if we know it, certainly dog is not the right species, we should go to the other species, with one exception. In the case of the proteins, monoclonal antibodies, without exception, predominantly you go for the primates only because monoclonal antibodies have no target either in the rat or the dog, so primates is the only species.
Thank you, sir. I think I have time for one last question. The question is, what is the role for safety pharmacology in impurity qualification studies?
Okay. Very, very good question. I think I would say not very much, but having said that, at least I would do on the impurity, we should do a hERG test. which I didn't discuss because of lack of time. A Herc test is a very simple in vitro test. We can do that. But generally, when you are given, when you are, again, these days, modern days, again, let me back off and explain for those who are not familiar. Impurity qualification applies only for the small molecules. Large molecules are highly purified proteins and monoclonal antibodies. Impurity is not an option, is not a question there. In the case of small molecules, these days, even without I would say least technology, we can actually get 98% pure. Most drugs are under development, they're 98. Bulk of the marketed drugs are 99.5% pure. So the amount of impurity that is there is 0.1 to 0.5, somewhere in that range. So the ability of that limited quantity to produce a systemic safety pharmacology, cardiovascular effect is quite remote. So it is unlikely any of the impurities can produce. However, if you do suspect, if you have any suspicion, don't ever waste your time, just do it. That's my recommendation.
Thank you, sir. Thank you for patiently answering all the questions. And, it's been a wonderful session. Thank you very much for your informative talk again, Dr. Rao.
Thank you very much, Doctor.
So we come to the fag end of the session. It's been a pleasure chairing this session. I think we've had 3 exceptionally wonderful talks from all the speakers, very informative across the board in terms of the content. I thank the participants, the audience, for their active participation. I think there have been a number of questions, and I'm sure that if you have more questions, please send it to the folks at DNDi, and they would be happy to pass them on to the speakers and get you a response. Personally, I'd like to thank DNDi and the Venture Center team for giving me this opportunity. It's been a pleasure hosting this session. Thank you very much. Soman, over to you.
Thank you, sir. Thank you to our session chair and to all 3 speakers for this very engaging and informative session. So appreciate all your support, and thank you for helping us to organize this and bring it to the larger crowd. And with that, I would also request all the participants, if you could stay over for a while. There is a link in the chat that you can, a feedback form. If you can kindly fill it, that would be very great, and we would appreciate your help. So with this, thank you, everyone, and hope to see you for the next session. Thank you. Thanks a lot.
Thank you. Have a good webinar series. Stay safe. Bye-bye.
Bye.
Thank you, sir.